Hey everyone, welcome back. In this video, we are going to continue with our notes on DNA, the introduction to DNA. And this time we're in section 4. And in section 4, we're going to be talking about the anti-parallel nature of double-stranded DNA. And notice this is going to take up 2 of these blocks here. So essentially 1/4 of the mega sheet is where we're going to draw and label the double-stranded DNA. Let's get started. I'm going to recommend you wait until the end of the video or until I say stop the video and draw now, because if you try to draw as I'm drawing, you may run out of space. So what we have drawn here is just a nucleotide, and based on the fact that I did not draw an OH group here at carbon number 2, now labeled for you, means that I'm going to be drawing DNA. So right here, had there been an OH group, this would have been a ribonucleic acid monomer. Instead, I've drawn DNA. And I drew this OH group here at carbon number 3 because it's going to be important when we add the next nucleotide. So I'm going to go ahead and draw another nucleotide. And so now we have 2 nucleotides getting ready to be joined together. So the other thing I did not draw was on this phosphate group, there are oxygens, there are hydrogen, there's other molecules here, but we simplify it by drawing it just as a P with a circle around it. Now, in the process of dehydration synthesis, that is, we're going to remove water to join nucleotide number 1 to nucleotide number 2. And so when we do that, we're going to do that here at this area along what we call the sugar phosphate backbone. So right here is where we're going to start breaking off molecules. Right here, we're going to start breaking off molecules as well. And it's here that we're going to remove water for that dehydration synthesis process. So we are going to remove 2 H's and an O, and what we're left with is a shared oxygen, here drawn in red, is a shared oxygen between this nucleotide and this nucleotide. That shared oxygen right here, now highlighted in yellow, is what is known as a phosphodiester bond. And so it's that shared oxygen that represents that phosphodiester bond that we made by dehydration synthesis between these 2 nucleotides. Now I'm going to add one more nucleotide. And so this is a trinucleotide. Tri means 3. And so what you see here are 2 phosphodiester bonds, one right here, one right here. And this was also made through dehydration synthesis. DNA is described as double-stranded and antiparallel. And antiparallel is just a fancy word for saying that this strand here is going to be upside down compared to the second strand we are going to draw over here. I would suggest at this point, go ahead and pause the video and draw this trinucleotide strand. Welcome back. So, we are going to draw the next strand of DNA, the second strand, what we call the complementary strand of DNA, over here on this side. And one of the things that I find is helpful to students is if you turn your paper upside down. By turning your paper upside down, it will help you draw, rather than trying to draw the same strand upside down, it's easier to draw if you draw it right side up. So flip your paper over and start here at the nitrogenous bases. So at the nitrogenous bases, we would have NB, and there would be a hydrogen bond between them. 2 hydrogen bonds if it's adenine and thymine, 3 hydrogen bonds if it's guanine and cytosine. So from here, I like to draw to carbon number 1. So I just take my pen and I'm going to draw here to carbon number 1. It does take some practice to draw this, but luckily, most of the time, all you have to do is identify it from a picture. But it is helpful to draw it out. So what I do is I draw carbon number 1, and then I draw directly parallel with that. So I'm going to go straight across here, and I'm going to draw carbon number 4. And I find that this helps me orient and get my pentagon into a roughly pentagon-ish shape. Now I'm going to draw my oxygen. I draw it up here, and then I just connect these two lines. Then I'm going to go ahead and draw the rest of the pentagon. Okay, not the world's best pentagon, but hey, this is an AP art. So from here, I'm going to draw that carbon at number 5, and then that phosphate group. And so I have the beginning of my antiparallel strand. So once again, I start with that nitrogenous base, NB, which is going to have some hydrogen bonds between them. Let's draw 3. So this is a hydrogen bond, this is a hydrogen bond, this is a hydrogen bond, meaning that's cytosine and guanine. Then I go to carbon number 1, carbon number 4, and in between them, I'm going to put the oxygen. Use that as my reference to make my pentagon shape. And then I got to add carbon number 5 that has a phosphate group attached to it. Now, I'm going to add my phosphodiester bond. And remember that phosphodiester bond is between the 3rd carbon. So if this is carbon number 1, then that's 2, 3, 4, 5. Here is 1, 2, 3, 4, 5. So my phosphodiester bond is going to be right here. And I do have to draw it a little smaller because I didn't quite make the space for it. But right here is that phosphodiester bond. Hopefully you did a better job than I did. All right, last one. Go ahead and pause the video now and try to draw this double-stranded DNA. Welcome back. Hopefully you have something that looks like this. It's a double-stranded DNA molecule with hydrogen bonds indicated in between those nitrogenous bases. So double-stranded means that the 2 strands are held by these hydrogen bonds between the nitrogenous bases, and the phosphodiester bond is along what we call the sugar phosphate backbone. So we go phosphate, sugar, phosphate, sugar, phosphate, sugar. So this is the backbone. It is the same in all organisms on Earth. Every organism has the exact same sugar phosphate backbone. And then the hydrogen bonds are what hold the nitrogenous bases together. And we call DNA antiparallel because this one has to be upside down compared to this one. So antiparallel is going to also help us in identifying the 2 strands of DNA because now we can label the 2 strands of DNA. So I'm going to zoom in now to show you how that looks like. When you take a look at this first nucleotide that we have drawn on the strand, you'll notice that going from top to bottom, the first carbon we encounter over here is carbon number 5. And so we don't count the phosphate group, we count the carbon itself. So on this strand right here, going from top to bottom, so starting up here as we work our way down, it would go 5, 3, 5, 3, 5, 3. But take a look at the other strand. It's upside down compared to the first strand. So over here on the other side, you'll notice that as we go from top to bottom, because it's upside down, this carbon over here is actually starting with the 3, with the 3rd carbon. So this one goes 3, 5, 3, 5. So if we're going to label these 2 strands of antiparallel DNA, we would label this side over here as the 5 to 3 prime strand. And this is the way we write it, 5, 2, 3. In other words, from up here, the first carbon we would encounter is number 5. The antiparallel strand over here starts with the 3, 5, 3, 5, 3, 5. And so this strand over here would be labeled as the 3 to 5 strand. This is going to be very important when we talk about DNA replication in the next lecture. I hope this video was helpful. Thanks for watching. Bye.